Welcome to Have You Read It, the channel where we bring books to life, one summary at a time. Today we're exploring Walter Isaacson's The Codebreaker, Jennifer Doudna, Gene Editing, and the Future of the Human Race. This book isn't just a biography of a brilliant scientist. It's a thrilling detective story about a revolutionary discovery, a scientific race filled with rivalries and collaborations, and a profound exploration of the ethical dilemmas we face as we gain the power to rewrite the code of life. It's a story of CRISPR, COVID, and the dawn of the life science revolution. Our story begins in Hilo, Hawaii, where a young Jennifer Doudna felt like an outsider. Her blonde hair and blue eyes set her apart in this Polynesian town. This feeling of being different fueled her curiosity about how she fit into the world, a curiosity that would later drive her scientific pursuits. A pivotal moment in Doudna's life came when her father, a literature professor, gave her a copy of James Watson's The Double Helix. This book, a detective story about the discovery of DNA's structure, ignited Doudna's passion for science. It made her realize that science wasn't just about men in lab coats, but an exciting quest to unravel the mysteries of nature. The foundation for this quest had been laid a century earlier by Charles Darwin and Gregor Mendel. Darwin's observations of finches in the Galapagos Islands led him to the theory of evolution by natural selection. Mendel, a monk tending his abbey garden, discovered the basic laws of heredity through his meticulous experiments with pea plants. Their combined work gave birth to the idea of the gene, the unit of heredity. The next step was deciphering the structure of the molecule that carried the code of heredity, DNA. This quest led to an intense race between some of the most brilliant minds of the 20th century. James Watson, a brash young American biologist, teamed up with Francis Crick, a British biochemist at Cambridge University. Their rivals included Linus Pauling at Caltech and a team at King's College London, led by Maurice Wilkins and Rosalind Franklin. Franklin's meticulous X-ray diffraction images of DNA were crucial, but her contributions were initially minimized. Watson and Crick ultimately prevailed, unveiling the double helix structure of DNA in 1953. Doudna's own scientific journey began in earnest at Pomona College, where she initially struggled with chemistry but persevered. A summer job working in a biology lab sparked her realization that she could indeed do science. At Harvard, she was mentored by Jack Szostak, who shifted his lab's focus to RNA, a molecule she found far more interesting than its more famous sibling, DNA. While Doudna pursued her doctorate, the scientific world embarked on the Human Genome Project, a massive effort to map all the genes in human DNA. This effort, led by James Watson, was both collaborative and competitive. Craig Venter, a maverick biotech entrepreneur, challenged the publicly funded project. Ultimately, they joined forces to announce the mapping of the human genome. Back in Szostak's lab, Doudna learned two important lessons: take risks and ask big questions. Szostak's big question was, how did life begin? He believed that RNA, not DNA, was the key. RNA could not only carry genetic information, but in certain forms called ribozymes, could also act as a catalyst to spur chemical reactions. Perhaps, Szostak theorized, RNA was the primordial molecule that kick-started life on Earth. This became the focus of Doudna's doctoral work. Doudna's work required her to become a structural biologist, using X-ray crystallography to determine the three-dimensional shape of molecules. At Yale, she teamed up with Jamie Kate to unravel the structure of a ribozyme. Their work, painstaking and often frustrating, involved crystallizing the RNA and then using X-ray diffraction to create images. Ultimately, they succeeded, showing how the RNA folded in a way that explained its ability to replicate. After Yale, Doudna and Kate, who were now married, moved to Berkeley. There she continued her RNA work, exploring the phenomenon of RNA interference, a process by which small RNA molecules silence genes. She determined the structure of the dicer enzyme, a key player in RNA interference, which would later become relevant to her work on CRISPR. The stage was now set for the CRISPR revolution. This story began in the 1980s when a Japanese researcher noticed odd repeated sequences in the DNA of E. coli bacteria, but didn't pursue them. Francisco Mojica, a Spanish scientist studying salt-loving microbes, later found similar clustered repeats and became obsessed with determining their function. He eventually discovered they were part of an immune system that bacteria use to fight off viruses. The system, called CRISPR, could remember and destroy viruses that had previously attacked. In 2006, Gillian Banfield, a Berkeley biologist studying bacteria in extreme environments, reached out to Doudna, then known for her work on RNA interference. They met at the Free Speech Movement Cafe on campus and discussed the CRISPR sequences Banfield had found. This meeting sparked Doudna's interest in CRISPR. Doudna brought Blake Wiedenheft, a postdoc, and Martin Gienek, a crystallographer, into her lab to work on CRISPR. They initially focused on Cas1, a CRISPR-associated enzyme that played a key role in how bacteria acquired immunity to viruses. At the same time, Rodolphe Barangu and Philippe Horvath, food scientists at Danisco, were using CRISPR to improve yogurt cultures. Viruses are a major threat to yogurt-making bacteria. Barangu and Horvath showed that CRISPR could be used to vaccinate these bacteria against viruses. Their work highlighted the interplay between basic science and commercial application. Back at Berkeley, Doudna was experiencing a midlife crisis. She briefly joined the biotech company Genentech, but quickly realized it wasn't for her and returned to her Berkeley lab. Upon her return, Doudna focused on building a strong lab team. Key additions included Rachel Horwitz, a talented biochemist and future entrepreneur, and Sam Sternberg, a researcher intrigued by the big questions of science. Doudna and Horwitz founded Caribou Biosciences, a company to commercialize CRISPR-based tools, initially focusing on diagnostics. Their venture highlighted the growing intersection of academic research and business. In 2011, at a conference in Puerto Rico, Doudna met Emmanuel Charpentier, a French biologist studying the Cas9 enzyme, a key component of the CRISPR system. Their meeting sparked a fruitful collaboration. Back in Berkeley, Doudna, Gienek, Charpentier, and Krzysztof Czeliński, a postdoc in Charpentier's lab, worked together to unravel the precise mechanisms of CRISPR-Cas9. They discovered the essential roles played by the cRNA guide and the trace rRNA, and they figured out how to combine them into a single guide RNA. Their June 2012 science paper describing these findings revolutionized the field. Their triumph coincided with a similar discovery by Virginijus Šikšnis, a Lithuanian scientist. His work, though important, didn't fully grasp the essential role of the tracker RNA. The two teams presented their findings at a CRISPR conference in Berkeley. The Doudna-Charpentier presentation, showcasing their single-guide RNA, was a clear breakthrough. The stage was now set for the race to adapt CRISPR for gene editing in humans. Existing gene therapies had shown limited success, and new techniques such as TALENs were cumbersome. CRISPR offered a potentially revolutionary tool. The race was on. Doudna, lacking expertise in human cells, faced a formidable competitor, Feng Zhang of the Broad Institute. Zhang, a brilliant young bioengineer, had been working on gene editing with TALENs, but had not yet made significant progress with CRISPR. Zhang's story began in China, then Iowa, then Harvard, where he was mentored by George Church. Church, a legendary geneticist, would soon become Zhang's rival in the CRISPR race. Zhang learned about CRISPR in early 2011 and began working on it in secret, even from his mentor George Church. He teamed up with Luciano Marafini, a microbiologist, to explore CRISPR. Back in Berkeley, Doudna knew that adapting CRISPR for human cells was the next big breakthrough. She brought Alexandra East, a grad student from the Broad Institute with experience in human cells, into her lab. East's experiments, combined with Martin Gienek's single-guide RNA, proved that CRISPR could edit human genes. Meanwhile, Zhang, working with Marafini, was still struggling. He had not yet figured out the full role of the tracer RNA. Then, in June 2012, he read the Doudna-Charpentier paper, It sparked him to action, and he adapted their single-guide RNA to work in human cells. Zhang and Church published their papers showing CRISPR working in human cells in January 2013, a few weeks before Doudna's paper appeared. This ignited a bitter rivalry, especially between Zhang and Church. The next race was to commercialize CRISPR. Doudna, Church, and Zhang initially explored forming a company together, but rivalries and disagreements intervened. Charpentier formed CRISPR Therapeutics. Doudna and Harwitz focused on Caribou and its spin-off Intellia, and Zhang and Church joined Editas. Doudna's relationship with Charpentier, strained by differences in style and Doudna's drive for recognition, cooled. They shared some major scientific prizes but drifted apart. While the companies were forming, Eric Lander published The Heroes of CRISPR, an article many saw as minimizing the contributions of Doudna and Charpentier. This fueled a backlash in the scientific community. The rivalry between Doudna and Zhang intensified with the battle for CRISPR patents. The patent office granted Zhang the first patents, sparking outrage from Doudna. The case went to court, with arguments focused on whether adapting CRISPR for human cells was an obvious or inventive step. The courts ultimately upheld Zhang's patents. Despite the patent disputes, CRISPR therapies began to show remarkable promise. Clinical trials were underway for sickle cell anemia, cancer, and blindness. While traditional scientists pursued these therapies, a new breed of biohackers, led by Josiah Zayner, sought to democratize genetic engineering. Zayner's public self-injection of CRISPR highlighted the ethical dilemmas of widespread access to this technology. Concerns about the misuse of CRISPR by bioterrorists led DARPA, the Pentagon's research agency, to fund research on anti-CRISPR systems, natural mechanisms that disable CRISPR. Doudna's lab became involved in this effort. The ethical debates surrounding CRISPR have a long history. Scientists in the 1970s gathered at Asilomar to discuss the safety and regulation of recombinant DNA. Similar discussions have surrounded gene therapy and pre-implantation genetic diagnosis. Doudna's own ethical journey began with a nightmare about Hitler using CRISPR. She convened a conference in Napa Valley to discuss the implications of germline editing, inheritable changes to human DNA. The group called for a prudent path forward. The ethical debate was disrupted in November 2018 when He Jiankui, a Chinese biophysicist, announced the birth of the first CRISPR babies, twins whose genes he had edited to make them resistant to HIV. His actions sparked global outrage. Doudna and other scientists convened in Hong Kong for a summit on gene editing. He Jiankui, originally not invited, was asked to present his work. He defended his actions, but was widely condemned. The CRISPR baby controversy highlighted the need for ethical guidelines. Doudna and others have wrestled with questions of individual choice versus societal good, treatment versus enhancement, and the potential for increased inequality. The COVID pandemic has accelerated the life science revolution, pushing scientists to collaborate and openly share research. CRISPR-based diagnostic tests have been developed, highlighting the convergence of biology and technology. The race for a COVID vaccine highlighted the importance of RNA. New vaccines, using mRNA, have shown remarkable success. This achievement showcases the power of basic research and the unexpected ways that scientific discoveries can be applied. Scientists are now pursuing CRISPR-based cures for viral infections. These therapies, though still experimental, offer a potential long-term solution to pandemics by directly targeting viruses. The Cold Spring Harbor CRISPR Conference in 2020 showcased both the scientific advances and ethical dilemmas of gene editing. It also highlighted the growing diversity of the field, but the need for more inclusion. The awarding of the 2020 Nobel Prize in Chemistry to Doudna and Charpentier celebrated the CRISPR revolution and the dawn of the life science era. The Jefferson conundrum, as Walter Isaacson poses it, asks how we should balance respect for a person's achievements with contempt for their flaws, especially when those traits are interwoven. This was brought to the fore when James Watson, after decades of scientific achievement, made reprehensible comments about race and intelligence, leading to his exile from the scientific community. His son Rufus's struggles with schizophrenia added a poignant layer to this story. Doudna's own evolving views on Watson highlighted the complexities of balancing scientific greatness with personal failings. Thanks for watching. Make sure to give this video a thumbs up and hit that subscribe button so you never miss our next book dive.